Hello class and welcome back. We're starting our third module and the start is on chapter 14, which is on nervous system tissue, where we start with a brief introduction in the anatomy of the nervous system. So how do we organize your nervous system? We organize it into two parts, your central and peripheral nervous systems. The central nervous system includes your brain and spinal cord, and the peripheral nervous system includes all of the cranial nerves coming off of the brain, the spinal nerves coming off of the spinal cord, and all of the ganglia, which are clusters of neuron cell bodies. The functional organization, we can also characterize the nervous system by how it functions, and we divide the central nervous system and the peripheral nervous system into three general functions. They all collect information with receptors. These receptors, which can be located in the back of your eye, on your skin, in other internal organs, these receptors will detect changes in the internal and external environment and then pass this information on to your central nervous system, whether that's your brain or spinal cord. Then the second function of the nervous system is to process and evaluate all of that sensory information. Your central nervous system will determine the required response. So we collect information, we process it, we evaluate it, and then we respond to that information. The central nervous system initiates nerve impulses to effectors to react to changes in the body's environment. And these effectors can be glands, skeletal muscle, in order to initiate an effect. There are two functional divisions of the nervous system. The two functional divisions are the sensory nervous system, which takes in all the receptor information from external and internal stimuli, and then the motor nervous system, which will initiate a response from that sensory nervous system. So this is a great chart that shows the functional organization of the nervous system. If we divide the nervous system, we divide it into the sensory component and the motor nervous system component. And again, the sensory nervous system contains receptors. It will transmit any sort of sensory information from these receptors, either externally or internally, to your central nervous system, which is the brain and spinal cord. Within the sensory nervous system, we divide it into the somatic sensory and the visceral sensory. Your somatic sensory receives sensory information from your skin, your fascia, your joints, your skeletal muscles, and all the special senses. The visceral sensory, viscera has to do with organs. So your visceral sensory will receive sensory information from all of your internal organs from the viscera. So the sensory information contains the receptors, sends it to the central nervous system, and then your brain pretty much sends out a response to the motor nervous system. So the motor nervous system will transmit information from your CNS, your brain and spinal cord, to the rest of the body. It will send this motor information to the effectors. And these effectors, we name them somatic motor effectors or autonomic motor effectors. Somatic motor means this part of the motor system is the voluntary nervous system. So this will innervate skeletal muscle. It's voluntary, and that means you control this. You tell your arm when to move. You tell your leg when to move. The autonomic motor is the automatic part of the motor nervous system. This is involuntary. These are motor nervous responses that will happen without you telling them to. So this will innervate cardiac muscle, your heartbeat, smooth muscle, the muscles in your digestive tract, and all of your glands, which will secrete saliva, hormones, and other things. So this is another just great look at the functional organization of the nervous system, and you should have a really good understanding of this functional organization. Simply put, the sensory nervous system is the input, where it detects stimuli and transmits information from the receptors to the central nervous system, where it gets processed, and then your central nervous system sends out an output, a motor response, that will initiate and transmit information from the central nervous system to the effectors. The somatic sensory is sensory input that is consciously perceived from receptors in your eyes, ear, and skin. Visceral sensory is input that is not consciously perceived from receptors in blood vessels and internal organs such as the heart. Then the somatic motor response is the voluntary, consciously controlled motor response. Usually, most often, the effector is the skeletal muscle, and the autonomic motor response is the automatic motor response. So not conscious, involuntarily controlled. The effectors will be cardiac muscle, smooth muscle, which is found in your digestive tract, and other glands. So the sensory nervous system, we also call it the afferent nervous system. It will receive sensory information from receptors in the peripheral nervous system and transmit it to the central nervous system. And again, it's subdivided into the somatic sensory and the visceral sensory. The somatic sensory includes all your general senses, such as touch, pain, pressure, vibration. Proprioception is the ability of your body to detect where it is in relation to all parts of it. So your joints and bones and muscles have the ability to just tell where everything is in relation to each other. And that's what proprioception is. Visceral sensory are impulses from all of your viscera, your internal organs. The motor nervous system, we call it the efferent nervous system. Think of the effect. It will send impulses from the central nervous system to your muscles and glands. And again, we divide it into two systems. Somatic motor causes contraction of skeletal muscle. This is voluntary, consciously controlled. The autonomic motor regulates smooth and cardiac muscle, anything that's subconscious or involuntarily controlled. Then we'll get into a little bit of the tissue of your nervous system. So we'll look at the cells, and that's what cytology is. It's the study of cells. There are two distinct types of cells within your nervous system. You will have neurons, which are the main nerve cell. These will be electrically excitable cells that will initiate, transmit, and receive nerve impulses. So they are the cells that are sending and receiving the signals that are passing through the nervous system. Then the glial cells are non-excitable, so they do not pass the stimulus through, but they will support and protect all the neurons. So here's a look at the neuron structure. A neuron has three main structural regions. The cell body, also called the soma, is the place that integrates, processes, and receives a signal from the dendrites. And the dendrites will be all these finger-like spiny projections off of the cell body. And the dendrites most often receive a impulse or a signal from another neuron. And then the axon is this long kind of extension that comes off of the cell body. The axon is connected to the cell body at the axon hillock, and the direction of the nerve impulse, also known as the output, will always leave the cell body and travel down the axon to the telodendria, which are the branches of the axon, and eventually to the synaptic knobs. And special nerve transmission will occur at the synaptic knobs in order to pass on the signal from this axon on to the next neuron. This is a microscopic image of a neuron. It's pretty fun to look at a neuron underneath a microscope. So the dendrites will be the short processes that branch from the cell body. They'll receive nerve impulses and they're carrying them to the cell body. The axons transmit these nerve impulses away from the cell body and transmit information to other cells. And the important region we talk about is the axon hillock, and that's the region where the axon connects to the cell body. Structures associated with the axons, and we spend a lot of time learning the structures on the axons because this will be the direction of the main impulse that will get sent on to another neuron. Axon collaterals are just side branches off the main axon. Telodendria are the fine terminal ending extensions. And then the synaptic knobs are the little tiny expanded regions at the tip of the telodendria. Some more neuron classification. Structurally, neurons are classified into three different types depending on the number of cell processes that are coming off of the cell body. A unipolar neuron has single short processes that branch like a T. So this is a unipolar neuron, shown at the top here. A bipolar neuron has two processes, one dendrite and one axon, and that's the second picture shown. And the multipolar, this is the most common type of neuron. It will have many dendrites and usually one single axon. And different types of neurons are found in different parts of the body, but the multipolar are the most common and the ones that you'll be studying most in anatomy and physiology. Functionally, neurons are classified according to their direction that the nerve impulse is traveling relative to the CNS. They will be sensory or afferent neurons if they transmit impulses from the sensory receptors to the central nervous system. And there'll be motor neurons or efferent neurons if they transmit impulses from your sensory nervous system to muscles or glands. Interneurons are a type of neuron that will be the mediator between the two. So these guys will facilitate communication between sensory and motor neurons. They will most often be found in the spinal cord. So here's a look at a skin receptor detecting some sort of sensation in the skin, whether it's hot, cold, pressure, pain, and that sensory information is traveling through a sensory neuron via the afferent transmission. It will travel through the posterior root ganglion, and a ganglion contains many cell bodies of sensory neurons. And then that sensory neuron will enter the spinal cord in the posterior horn of the spinal cord. It will synapse in an interneuron. So again, the interneuron is kind of the connector between the motor and sensory neurons. And then the interneuron will pass along that signal to a motor neuron. And the motor neuron will take the effect or the response through an effector or efferent transmission to the axon. And eventually, so this is going to a skeletal muscle. So let's say this woman felt some pain, her skin receptors felt pain, and then that response told her arm muscle to move to try to get away from that pain. So this is a great chart to review the structural and functional classifications of neurons. So feel free to pause the video and just take a couple minutes to view this on your own. So here are some review questions. Another term for the sensory nervous system is the afferent, and motor nervous system is the efferent. List the two divisions, each of the sensory and motor nervous system, and you can go back to those first couple charts that we went over previously that listed the complete functional divisions of the two. List the three types of neurons classified by their structure and function, whether it was the unipolar, bipolar, or multipolar, and which neuron is most numerous in the nervous system, and that was the multipolar neuron. Then we'll get to glial cells. They're sometimes referred to as neuroglia, and again, these are cells that help support neurons. They're smaller than neurons and they are capable of mitosis, so dividing, unlike neurons are. They physically protect and nourish the neurons. They're more numerous than neurons, and brain tumors are more likely to be derived from glial cells than neurons. So you'll hear of gliomas. That's a tumor of the glial cells or a brain tumor. These are the four types of glial cells in your central nervous system. You should be very familiar with all four: astrocytes, ependymal cells, microglial cells, and oligodendrocytes. And again, these four are just located in your CNS, so your brain and spinal cord. Astrocytes are the most abundant glial cell in your CNS. They form a blood-brain barrier, and we'll talk about how they do that. They have little tiny green perivascular feet that wrap themselves around blood capillaries. So they will only allow certain things to come out of the blood capillary into the brain tissue, and that's what we call the blood-brain barrier. Certain things can pass through the blood-brain barrier, like nicotine and some drugs. Astrocytes will also help regulate tissue fluid composition. They'll help regulate synaptic transmission. That's the transmission of electrical signals between neurons. They will form a structural network in your CNS. They'll replace damaged neurons, and they will assist in neuronal development. So again, this is the most abundant glial cell in your CNS. Ependymal cells are ciliated cuboidal epithelial cells that line the ventricles of the brain and the central canal of the spinal cord. In conjunction with other glial cells, the ependymal cells help to produce cerebral spinal fluid. This cerebral spinal fluid fills up the ventricles and the central canal and the spinal cord. The ependymal cells also form the choroid plexus, which helps to form the cerebral spinal fluid. Microglial cells are small cells that are movable, they're motile, they can move. They will wander through your central nervous system and exhibit phagocytic activity. That means they'll engulf any sort of cellular debris from dead or dying cells. So they're kind of the cleanup cell of your nervous system. And then oligodendrocytes are associated with your central nervous system axons only. And I want you to remember that. Oligodendrocytes are only in your central nervous system. They will wrap themselves around the axons like electrical tape wrapped around a wire, and they produce myelin, which is an insulator of electrical activity. Myelin will help pass along nervous system signals more quickly through axons. Neurolemocytes are also called Schwann cells, and these are like oligodendrocytes, except they are in the peripheral nervous system. So say that a couple times. Oligodendrocytes are in your central nervous system, and neurolemocytes are just in your peripheral nervous system. They do the same thing. They wrap themselves around axons like electrical tape wrapped around a wire. They produce myelin, these myelin sheaths, which is an insulator of electrical activity. And again, that just helps the electrical signal pass more quickly down an axon. This shows you how myelin is formed around the axon. Myelin will affect the ability of neurons to conduct nerve impulses, which we also call action potentials. You'll learn a lot about that in physiology. Myelination is the process of wrapping the axon with a myelin sheath. The myelin sheath is formed by neurolemocytes in your PNS and oligodendrocytes in the CNS. And again, it serves as insulation to help conduction speeds of these action potentials or the conduction of a nerve stimulus. Saltatory conduction describes how current or the electrical charge passes through a myelination axon, and it only passes through at the nodes of Ranvier. And the nodes of Ranvier are the spaces between these little blue myelin sheath packets. At the nodes of Ranvier, you have more concentrated voltage-gated sodium channels, and action potentials will be triggered only at these nodes, and they'll jump from node to node. And we call that saltatory conduction. Saltar means to jump in Spanish, I believe. So the saltatory conduction jumps between these nodes of Ranvier where that action potential will occur. And again, this will be much faster than conduction along unmyelinated axons. So the myelin sheath helps the conduction of this electrical signal. Conductive velocities of axons vary widely among neurons. The rate of the impulse propagation is determined by axon diameter and the presence of a myelin sheath. So the larger the diameter, the faster the impulse. And if the axon is myelinated, that will also dramatically increase impulse speed. So impulses will move fastest along axons that are larger in diameter in comparison, and also ones that are myelinated. Neurophysiology talks about the physiology of the nervous system. Neurons are highly irritable, meaning they will respond very easily to stimuli. Action potentials or nerve impulses are electrical impulses that are carried along the length of axons. They're always the same regardless of the stimulus, and they are the underlying functional feature of the nervous system. Action potentials, and you'll go over this a lot in physiology, but we're going to briefly introduce it here in anatomy. An action potential is a brief reversal of membrane potential with a total amplitude of 100 millivolts. Action potentials are only generated by muscle cells and neurons. And when we talk about a brief reversal of membrane potential, we're describing that the inside of these neurons, there's always a negative charge on the inside of these neurons. And when a neuron gets stimulated, the charge changes by a total amplitude of about 100 millivolts. And that change in membrane potential or charge is what generates an action potential. They do not decrease in strength over distance, and they are the principal means of neural communication, and action potential in the axon of a neuron is a nerve impulse. So it's just the way that signals get traveled down axons. Multiple sclerosis is an autoimmune disease. So an autoimmune disease, whenever you see that word, that means it's a disease where the body attacks itself. MS affects mainly young adults and for some reason women more than men. The symptoms of it are visual disturbances, weakness, loss of muscular control, and urinary incontinence. So the inability to control urination. Nerve fibers are severed in MS, and myelin sheaths in the CNS become non-functional scleroses, which are like hardened patches. So this will shunt and short circuit nerve impulses from occurring. So basically your nerve fibers become severed, the myelin sheaths become hardened scleroses. So the nervous system loses the ability to conduct nerve impulses. So people with MS will lose the ability to control their muscles, their vision, urinary incontinence, really any place in the body where neurons control some sort of muscular activity. Nerves then, so what is a nerve? When we learn about all the nerves that you're learning in lab, it's actually a cable-like bundle of parallel axons, and it's surrounded by 3 connective tissue wrappings. The endoneurium will wrap around each axon. The perineurium wraps around individual fascicles, and the fascicles are made up of axons. And then the epineurium is around the entire nerve. So here's a look at the entire nerve. Again, and a nerve is just a cable-like bundle of parallel axons. The nerve is made up of fascicles, which are surrounded by the perineurium, and fascicles are made up of axons, which are surrounded by the endoneurium. Synapses. When we talk about synapses, this is just a specialized junction between one axon and another neuron. So whenever an axon ends at the synaptic knobs at the end of its telodendria, that axon has to somehow send along the nerve impulse to the next neuron that's on the other side of it. A typical synapse consists of the presynaptic neuron, where the nerve impulse will end at the axon terminals, the postsynaptic neuron, which will be the nerve that receives the nerve impulse and will continue to pass it along, and the synaptic cleft, which is the jelly-like or fluid-filled space between the two. Chemical synapses are specialized for the release and the reception of neurotransmitters. They're typically composed of 2 parts, the axon terminal of the presynaptic neuron, which will contain synaptic vesicles, which are just kind of holdings of the neurotransmitters themselves, and the receptor region on the dendrites or cell body, the soma of the postsynaptic neuron. The synaptic cleft is a fluid-filled space separating the pre- and postsynaptic neurons. It will prevent nerve impulses from directly passing from one neuron to the next. The transmission across the synaptic cleft is always a chemical event as opposed to an electrical one. So nerve impulses pass through individual neurons electrically. So there's a change in voltage in each neuron axon. But to move this passage of a nervous signal from a neuron to another neuron, we have to release chemicals, and those are the neurotransmitters that get released across the synaptic cleft. This ensures unidirectional communication between neurons, so it allows for a lot of control within the synaptic cleft. So how do we transfer this information across the synaptic cleft? Well, the nerve impulse will reach the axon terminal of the presynaptic neuron. The neurotransmitter will cross the synaptic cleft and bind to receptors on the postsynaptic neuron, and then the postsynaptic membrane permeability changes, causes another excitatory or inhibitory effect on the postsynaptic membrane. A neurotransmitter is the chemical used for neuronal communication with the body and the brain. So these will be released between pre- and postsynaptic neurons. There are 50 different types of neurotransmitters that have been identified, and they are classified how they're structurally formed chemically, and what their functionality is. There's basically two types of neurotransmitters, whether they are excitatory or inhibitory. So whether they will cause an excitatory response to continue or an inhibitory response. Excitatory neurotransmitters will cause depolarization. So that means another action potential will occur in the second neuron that it's trying to excite. And inhibitory neurotransmitters will cause hyperpolarizations, meaning they'll keep the neuron at a hyperpolarized state, which means it's harder to get an action potential started. Glutamate is an example of an excitatory neurotransmitter, and the GABA protein and glycine are inhibitory neurotransmitters. That's the end of this side. Thanks for listening, guys. Come back for chapter 15 when we talk a little bit more about the brain. Thanks.